really great. And that matters too. My name is Mark Holey. I organize this with Crossroads, and our team at Crossroads includes Sam Coyen, Hagen Haringa, and Lynn Mickelson. And we get Zoom support from Door County Library, and Laura Kayakon is on at the other end running that. And also Maggie Beam helps us there. So it's a great team. It's worked out well. This is our 8th season, if you can believe it or not. So it's coming along very well. So before we get started, I do want to say, let's see if I can do it. Yeah, I got to mention the rest of the talks. So February 12th, we're going to hear about walleye up on the top. It's kind of covered up there, but Jason Bregeman, who's now the fish manager for Southern Green Bay, he manages for the DNR. He will talk about the walleye population. I was just talking to a gentleman earlier saying, what happened this last year? Walleye was off. Well, at least we'll get the biologist's perspective on it. And you can ask all the questions you want. And he'll take us back to when walleye collapsed, when stocking started, the whole story. That's what he's told me he'll be able to present. And then on February 26th, a former fish chief or bureau director of fisheries management for DNR who was worked for the organization when the salmon program got started. He was a field biologist in Green Bay. He eventually moved to Madison to be the Great Lakes sport specialist, and then became the fish chief as well. So he's been the bureau director. And that was back before computers. So he's going to give you a firsthand account of what he had to go through, or what the organization had to go through to bring salmon to Wisconsin. In fact, that picture there shows the largest fish is the first year the cohosts were stocked in Wisconsin. And then the smaller one was the second year. And that's what he has to show for that. And then lastly, we have Dr. Dan Eisenman that will talk about smallmouth bass. Karen's going to talk about telemetry, and so is Dr. Eisenman. But the big question there was, if you catch fish over one spawning bed and then transport it somewhere else, what happens? So he has fish that are tagged with these radio tags, where he's caught some in Sawyer Harbor right during spawning. And he put them in several spots, but I know he put some on Egg Harbor. And what happened to them? Did they all run back to Sawyer Harbor or not? Well, that's a very interesting question, not only scientific, but also practical for all you fishermen, right? And that'll be very good as well. Getting back to Karen, this is her 3rd appearance here. And she's very popular because she has a lot of good stuff to say. But I would have introduced her twice already. I think a lot of you have probably heard that. You've heard that she's the director of freshwater research at the Shedd Aquarium since 2016. That she leads a research program focused on understanding how environmental change and human activities affect native migratory fish. And that she relies on an amazing group of volunteers that help collect sucker data on a good number of Lake Michigan tributaries and a few in Lake Superior. That's the scope of her project. Let's see. But I want to tell you something that you don't know about her, because the rest of it, you're all pretty familiar with Karen. And this is just something that, for those of us that work on Lake Michigan, the Fishery Commission relies on advisors. Now, these are people in the field that are out there and know the system. So they have discussions with fish managers, but they also have an organized advisor. Now, if you came and watched the Fish Thief about sea lampreys, they talked back in the 30s and 40s and 50s that they had an advisor, and actually that started that far ago. And the cool thing about Karen is that now she's the public at large advisor for Illinois. Usually they have one for a sport fishing representative, a commercial fishing representative, and if you came to the movie, you knew that Dennis Hickey from Door County was here as a sport rep, and Lee Hosh was here as as the commercial rep, and Lee Hosh was here as a sport rep. Well, there's a third one, and that's a person at large. So now, recently, Karen's been designated as that person at large for Illinois. So it shows her status in the Great Lakes region that includes support by the states. And she will be some of the eyes and ears of all the field work she does to help inform the commission when they coordinate fisheries management, but also how they select this sea lamprey streams too. They run that whole decision matrix by their advisors as well. So we're really lucky. The other thing about Karen, and you notice this is a stage, she actually put this on her Facebook page, so the stage with the sucker right in the bottom there. But she's got a huge following. You know, once Lynn Mickelson produces the posters and stuff, then I start distributing them and all that kind of stuff. And I went down to Lawless Coffee and went in with there's a big poster and a little one because some don't like the big ones. And I always ask, can I put your poster up? And I'll this poster up. And these two, from my perspective, younger women were behind there. He says, well, what's your poster about? And I said, well, it's good fish talks and all this stuff. Before I got through a couple sentences, both of them said, oh, Karen's back. Yay. Yeah, unsolicited. And even this Wednesday night at the Plan Commission, the mayor started before the small talk before this, the mayor says, hey, Karen's coming this week, I know that. So it's a huge following, and it's very impressive. So with that, I'm going to just then say, for the ever popular Karen Murchie, take it away.
Thank you for the introduction, Mark, and thank you so much to Crossroads for having me here. Thank you all for coming out in the cold weather to hear me talk about suckers. I've been at the aquarium now coming up to my 10-year anniversary, and the whole time I've been studying suckers and bringing people on board to become fans of these fish. So how many people have heard of suckers before? Great. You're all prepared for learning some more about them, but I don't have to do the extra hard work of convincing you why they're so awesome. You may or may not know that suckers are the most abundant group of migratory fishes in the Great Lakes. So there's a number of species that fall under the sucker family. The most common and most abundant are the white suckers, followed by the long-nosed suckers. And then you, of course, have the various red horse species as well that are also under Catastomidae, but both by biomass, just the size of them, they're pretty big fish, around 18 inches. And the numbers, they do make up the largest group of migratory fishes. And there's a lot of value that these fish are adding in the ecosystem. Probably, do most of you know about suckers just because you've seen them in the spring in the tributaries spawning? Is that, or what are, shout out any other reasons that you're familiar with suckers?
I tried eating some once and they're really bony. I lost more calories trying to.
Yeah. So some people have eaten them, others have caught them, seen them in the creeks. But I like to refer to them as the wildebeest of the Great Lakes. So a lot of the times when we're thinking about migratory animals, we think birds, we think butterflies, and then if we're thinking about fish, some of the most common fish species are, people start thinking about the Pacific salmon and how awesome they are. But suckers, they deserve the same level of clout. Because they're making these amazing migrations. They're in all 5 of the Great Lakes, and then in the springtime, they're moving from the lakes themselves into tributaries, creeks, and rivers of all sizes to spawn. And you can have, in a tiny little creek that is only 1.5 feet deep, you can have over 1,000 suckers. And then some of the really large tributaries, you're getting tens of thousands of suckers. Even one tributary of Lake Superior, some researchers went out and they netted every single fish. And with both white suckers and long-nosed suckers, they had over 82,000 suckers in one river. Multiply that around all the Great Lakes and all of the tributaries, large and small. and that is a lot of fish. And so, of course, they're coming into the tributaries to spawn, and reproducing is really important for carrying out, sustaining the populations. A lot of the fish that people love to catch and eat rely on eating suckers in a number of parts of their life stages. A lot of the trout, particularly in the Upper Peninsula of Michigan, the bulk of their calories that they're going to get in a year are eating sucker eggs during those spawning migrations. That is the biggest bulk of their caloric intake in a year. And then, of course, even the northern pike, they'll move into wetlands to spawn. And even the tiny baby, newly hatched pike will start eating the even smaller baby suckers at a really young age. And then, of course, too, you've got avian predators that are going to be feeding on the suckers as adults while they're in the creek spawning. I've seen both eagles and osprey take suckers, and then recently found out about some super cool footage from a tributary in Minnesota. They had a trail camera out, and they were documenting wolves coming and harvesting out the suckers from the creeks as they were coming in. So all sorts of nature is really cued in on these migrations and taking advantage of it. And then, of course, too, other fish will feast on suckers, particularly when they're really small. And people have used baits and even lures that look like suckers to catch fish. So also, while they're in spawning, their eggs, their sperm, as well as some excretory nutrients come out, and it basically acts like fertilizer. Things like nitrogen and phosphorus that you would put in your garden, they're finding that they're adding those nutrients to the creeks as well. And essentially, they're kickstarting the food webs. They're acting like little gardeners of the creeks, adding this nutrients, and the algae grows faster where the suckers have been. The little tiny baby aquatic insects in their larval stage while they're in the water, things like caddisfly larvae, really popular food for trout. They grow bigger where the suckers have been. And if you're a trout, you can eat a larger caddisfly larva. That's like upgrading from a hamburger to a Big Mac. So I say if you love trout, you have to think a sucker. But also if you love birds and bats, you should think suckers too, because those aquatic insect larvae, if they don't get eaten by fish, hatch and are food for birds and bats. So how many people are sucker fans already? I'll be on slide 2. These fish can live a long time as well. Long-nosed suckers, the oldest, have been aged to 40. White suckers in their 20s. And probably, even if there was more research, I bet we'll find that there are older suckers as well. Within that Catastomidae family, there are some species that they've recently discovered live into their hundreds, the buffalo fish. So it's possible. These are just a really understudied group of fish. We have so many more discoveries to make about them. And what's cool, when they spawn, they don't die. So they're called iteroparous. So they can start spawning at age 3 or 4. And then year after year, if they've had a good winter, they've been able to, have enough energy to avoid getting eaten, find the food. They can put energy into making either eggs if they're a female or sperm if they're male, and be ready to spawn again. And year after year, they can do that. They don't die unless, it's their time. They typically don't die, and they have a pretty low spawning mortality rate. So that's pretty cool. And I think I already mentioned too, all that kickstarting of the food web is a really important piece. So that's one thing we know about the suckers. But again, there isn't a lot of knowledge to date because they're not typically a fish that people go out and fish for. They have historically been used as food by the Ojibwe. The Ojibwe have a moon phase. They have a sucker moon because they're important to them. And a lot of people have fished for them as well. I've heard, they also are supposed to taste good. I hear more people will pickle them as well. I have yet to try them. So far, I've become quite attached to them. But after one of the fish tales I gave a number of years back, I had somebody tell me that they used to have a commercial harvest for them. And then the harvested suckers would be sent to Canada for processing, and then they send them back here, and it was turned into sort of like a sloppy Joe, sloppy sucker mix and fed in cafeterias, but. I think that would be an interesting piece to follow up on, so it's pretty interesting, but yeah, again, most of the time, like our state agencies are learning about a lot of the fish that people are fishing for. And rightfully so, because there's a lot of management for that. But for our most abundant group of migratory fishes, beyond some of the stuff I told you, we can visibly see them moving in these mass migrations into the tributaries. We see things eating them. That's sort of about where the knowledge goes. There's some work done more in like their susceptibility to contaminants and some contaminated rivers, but where do they go when they're not spawning? All these types of things are giant gaps. And that's not so good, because if you're trying to think about their populations and watching how they're doing, well, you need some of this information. You need to know what kind of habitats are they relying on outside of spawning season. If we start to see their numbers declining, what might be going on? We're not really paying a lot of attention to that. And so also, if we don't have a lot of information about them, it's hard to share their stories and connect people to how awesome they are and get people to care about them. So that's really important as well, because when you don't care about something, you don't protect it. And unfortunately, these are some things that can happen. I used to not show slides like these, and I started last year because I think it's important. I don't think I'll cry tonight about it, but working with these fish, it's you get really attached, you care about them, and when you see some of the things that people do to suckers, these are all pictures taken from Sturgeon Bay, mostly here at Big Creek, but one another creek, Samuelson Creek nearby too, where people have done some pretty awful things. And so when you have big piles of fish being removed, not for harvest, not for any reason other than some people deem them as trash fish. And that is a category that no native species should be put into. Even in terms of harvest regulations through most of the Great Lakes region, there's even very little set for harvest regulations, or the number's super high. Like you can take 600 suckers a day. And so those kinds of things, and just our lack of storytelling about them kind of gives an impression, well, maybe they're not that important. There's a lot of misconceptions that, oh, the suckers are eating the steelhead or rainbow trout eggs. Well, they might hoover up a few, but by and large, those steelhead are benefiting immensely from these animals. So if we can learn more about them, we can share their stories and connect more people to care about them as well. So a lot of the things I'm trying to answer are questions of like, where do these fish go outside of spawning season? So we know that they're coming into these creeks to spawn, but are the same fish that are coming in to spawn together, do they come back to that same creek year after year? Do the fish that spawn together hang out all year round? Are they all BFFs and, just go to places together? And I don't know. And then for each individual sucker, how different are they? Do they all act the same way? Is there like people, some people are homebodies and some love to travel? Is that what we see with them as well? Which is pretty common for a lot of the fish species that we've studied collectively, whether they're freshwater fish or marine fishes. We often see quite a lot of individual variability. So these are some of these things that are really important questions to ask. And one way of answering some of those questions is through telemetry or tagging and tracking, just very simply.
Click on that okay up there.
Oh.
This has been driving me crazy.
Thanks.
There.
Thank you.
So fun.
I'm so wrapped up in my storytelling. So by tagging and tracking, we can start to answer some of these questions. And all the photography that you're seeing in here are all pictures that I've taken, unless I'm in it, then I didn't take it. Although I am pretty good at selfies. But these are a couple of white suckers, that are getting ready to spawn. That was from up in Fish Creek. Okay, so I'm gonna start with some really basic tags. How many people have a pet at home that is microchipped? A good number of people. My dog is, she's in the middle picture. I like to be able to get Fiji into my slides. So these tags, what's great about them, these microchips, is they have no battery in them, right? Could you imagine after like 5 years having to bring your pet in and they have to dig it out of like your animal and like put a new battery in? That'd be awful. They're really inexpensive as well. Maybe you're charged more to put one in, but they're only like a few dollars a piece, which is really nice because that also means you could tag a lot of animals. They can be really small, the smallest, like, the size of a grain of rice, and then you can get a little bit bigger, but either way, they're really small. You can tag a lot of different life stages of fish with them. You can tag all sorts of animals with them as well, terrestrial animals too. But they're used pretty commonly for fish and in both marine and freshwater systems. So yeah, again, what's nice, let's say your pet runs out of the house, gets lost, somebody can pick it up, bring it to a veterinary office. They use a wand, a handheld scanner to scan it, and each tag has its very own unique ID. They can match it up with, oh, that's so-and-so's pet, call the number, etc. Similarly too, every tag that we put into a fish has its own unique ID. We don't pick them all up and scan them, but instead we can install antennas into some shallower creeks or larger rivers. And as a fish swims in, it checks itself in, and it's really pretty cool. So it's nice too, it's really simple to put these tags in. Typically, well, for the suckers, we just put them into the open body cavity. So you just use a little needle, inject it in, similar to how it's injected into your pet. Some people put it in the muscle tissue. It just depends sort of on the biologist and what the questions are. A lot of all the lake sturgeon reintroduction stuff that's been happening, all the little baby lake sturgeon before they're let loose back out into the water, they're all tagged and tracked to see if they come back too. So it's used for a lot of questions in the Great Lakes to understand some fish movement, especially around restoration issues. So they're pretty cool. So this is a white sucker that got a pit tag, and then we can track where it goes when it swims back into a creek. So when I started this program, I'm like, I don't know what to expect if I tag suckers. How many am I gonna see? So between 2017 and 2018, working in Samuelson Creek, which is a tributary just across Sturgeon Bay here on the other side, a really small creek, mostly residents on either side. There's an impassable barrier just on the other side of the highway. Really easy system for me to work in. I thought, well, I'll get 300 fish tagged between those 2 years and see what happens. So had two antennas like in the middle picture that you can see, as well as a submersible antenna. So as the fish come in from Lake Michigan, well, and then Sturgeon Bay, and swim into the creek, as they swim over those antennas, there's a reader that picks it up. Oh, this is fish number 23-digit long number, the date and time, etc. And what you can see from the numbers that I've got up there for results, we were getting amazing return rates. So to that initial 300 fish tag, 100 in 2017, 200 in 2018, in 2019, 204 out of 300, 2020, 176, and then 2021, 143, etc. So well over 50% return rate most of the time, which is really awesome. And then I started to add fish with some acoustic tags, which we'll get into in a minute, and some already had pit tags and then some didn't, and I added them, so that's why you see these numbers jump to out of 347. But the big takeaway is, you know, that first question, does the same fish come back to the same location year after year to spawn? And we're starting to see just through this pit tagging technology, yeah, a lot of the times we're seeing the fish come back year after year to this one tributary. So that says something too, especially if you have a culvert, which culverts are everywhere, right? You're not gonna build a bridge over every tiny little creek. You usually have culverts to allow the water to pass through a small street or under a small street and allow fish in the water to pass through as well. Often over time, something can happen where you have erosion going on downstream of the culvert and it can become perched and have a bit of a waterfall, and then the fish can't always get upstream. So sometimes those culverts can be impassable and that can change access to spawning grounds. So these are some things that we can think about of like, okay, what happened if fish are coming back year after year to a spot, but there's an impassable barrier? This kind of information is useful to managers to say, okay, is an important tributary for this population of fish. We should try to do some restoration efforts to fix that culvert. Okay, so some of the big findings I started to reveal some of that seeing high spawning site fidelity. So that also means in the case of some of this wanton waste issue where you have people indiscriminately killing fish and throwing them up on shore, the damage that could be done to one tributary by killing, you know, hundreds of individuals each year, that could have some serious ramifications for that individual tributary and the health of it, not just for the sucker population, but for the aquatic invertebrates, the other fish, the birds, the algae, etc. So that's an important piece that we can start to connect. One thing that's really cool, looking at the data of the movements of the fish in the night, when it's not so easy to just go out and see what's happening, you see the males starting to have their little adventures into the creek. After 10 p.m., some individual males venturing into the creek. Are the ladies here yet? Checking it out? No, no ladies. By 2 in the morning, I've gone back out into the lake to wait. Next day, 10 p.m. rolls around. Are the ladies here yet? No. And you know, this happens for a number of days until the water temperature and flow conditions seem to be right. I know Mark mentioned that I've got a number of volunteers, some of them in the room, that help me figure out by having a number of locations with people helping me monitor when they see suckers showing up to spawn. We match it with water temperature and water flow cues. And we found with the first 7 years of data from the volunteers from a number of tributaries in Illinois, Wisconsin, and Michigan, both Lake Michigan and Lake Superior, water temperature is the most important cue. So they're waiting for that right temperature to move in. And then once that temperature hits, then you see all these fish move in, and then they hang out and stay in the creek to spawn.
What is the temperature?
It is, now I gotta remember what it is in Fahrenheit. Give me a second, my brain's,
That's
I'll
fine.
follow back up.
I got all night.
Yeah,
What do
you
you
know,
Celsius?
no, I'm trying.
Oh, I think you knew it in Celsius.
Yeah, I know, but give me I'll come back to it. I'm sorry, I'm totally like blanking on that. Just so I don't get it wrong, that would be embarrassing. This is my study. So what some new information hot off the press as of a couple weeks ago, I mentioned that these pit tags, other organizations, the Wisconsin DNR and others, like US Fish and Wildlife Service, are using pit tags to tag a lot of different fish species, but particularly a lot of interest with lake sturgeon, right? And so a number of rivers have pit antennas, large ones that can span the width of the river to pick up returning lake sturgeon that were stopped there from restoration initiatives. So in the Peshtigo River, if you're not familiar with where that is, I don't have a pointer, but the more western yellow dot, and then the other yellow dot within the Door Peninsula is Samuelson Creek, of course. A fish that I tagged in 2018 in Samuelson has been getting picked up in the Peshtigo River. They only found this out a couple weeks ago after being on a Green Bay coronation call and having one of the Wisconsin DNR biologists go, I never thought to ask you whether any of the unidentified fish that we've seen on our pit antenna array might be any of the suckers. And they sent me the list of unidentified tag IDs, and the third one in was this white sucker from Samuelson Creek, tagged in 2018. I had not seen it since. I had tagged it that year, but it's been hanging out in the Peshtigo River. They put the antenna in 2020. It hasn't been working perfectly every year, but in 2023 and 2024, it was picked up spawning in there. That's pretty cool. And I have to remember, it's been a long day, how many miles, I think it's 27 miles as the crow flies from Samuelson to where that antenna is by the dam on Peshtigo River. So that's as the crow flies, but as the sucker swims, that's quite a ways to go just to make that initial thing. So since I tagged that fish in Samuelson Creek in 2018 and never saw it again, did it just decide to stray over to the Peshtigo River and then stay there for a while, or had it come from the Peshtigo River into Samuelson Creek the year that I happened to start tagging and got tagged then? That's pretty interesting. Also, last year, one of my tag suckers from Samuelson Creek in 2017 was harvested in Big Creek right here. One of the Wisconsin DNR biologist kids had harvested suckers to take home to eat. They found the pit tag handily enough, knew that I was the only person pit tagging suckers, reached out to me and said, I think we found one of your fish. So that is another neat connector as well. So we hadn't, they ate the fish, not the tag. The tag is just loose, you know, as they clean the guts out, it just fell out. So we found it before that. But those little pieces of information are helping me put some other puzzle pieces together, like, okay, it's not just that when I tag those fish that maybe they died, or they were taking years off of spawning because maybe they didn't get enough energy. That there is some movement, there's some straying between systems, so they might be really, might prefer a spawning site for a long time, but then they might also have that flexibility to adventure off and try spawning somewhere else, which is really good to have that sort of resilience baked in, that you aren't so reliant on one tributary. I need some water. Because some of the tributaries too, of course, even just within Door County, some of them are really intermittently connected. So not every year, because they're really tiny creeks. I gotta drink. They might not have enough of the snow melt and rain in the spring to like clear away sand at the mouth of the creek. So maybe the fish can't get in, right? So there's some tributaries. Just on the other side of Rigida Sanctuary by Toft Point, there's like a little tiny wetland area. It gets a really strong run of long-nosed suckers when it's connected. When it's not connected and they can't get in, where do they go? Do they have another spot that they could go? So it makes sense that, again, that they have that flexibility, but it's really neat from these two pieces of information from the DNR, you know, putting together some of the travel history for these fish. All right, the pit tags are great for a lot of reasons. Again, they're inexpensive. You can tag a lot of fish. They're only like $7 a piece for the little bit bigger ones. Not too expensive. But you can't have a carpet of pit antennas out in the big waters of Lake Michigan. They don't work in that kind of a system. So this is where we have to turn to a different type of technology called acoustic telemetry. And basically, again, that's got a tag component to it. But these tags have batteries, and the batteries help signal or send out the signal, the acoustic signal that travels well through water to a listening device or a receiver that receives the signal. And these receivers, we're lucky that we've got a lot of them out in Lake Michigan. Green Bay is really covered. I'll show you a slide in a second. But essentially, those two components work together to help reveal where all sorts of fish species that we're interested in studying go. And anybody that watches some Shark Week shows, and they can tell you where sharks have been going, a lot of that is with acoustic telemetry. Some is with another fancier telemetry satellite as well, but a lot of that information comes from acoustic tags, and they're about $400 a piece. They're not cheap either. So that means you can't tag as many animals, especially not with limited funds. But what you can see in this slide here is the Great Lakes Acoustic Telemetry Observation System, which is a Great Lakes Fishery Commission funded project that is made up of collaborators from both the US and Canada, all sorts of agencies, state, federal, provincial, tribal, academic institutions, non-government organizations, all contributing infrastructure, those receivers to listen for the fish. So every yellow dot that you see on this map of the Great Lakes and even into the St. Lawrence is a listening station. And this hasn't looked like this since the Great Lakes tagging and tracking stuff started. Bit by bit, this has grown. It really started with more, okay, in Lake Huron and Lake Erie, where with some walleye studies that I was a part of back in 2011, is really a time when things really started to get going to look to see how much do these fish move? Because if you have state agencies and provincial agencies managing walleye populations and everybody setting harvest regulations, and you think, okay, these walleye just stay within like Wisconsin waters or Ohio waters or Ontario waters. This is really important to know how much are they mixing. So this is really that impetus to start tagging and tracking not only walleye, but then starting to answer questions about lake sturgeon and lake whitefish, and then smallmouth bass and northern pike, and then eventually suckers. And because of all this infrastructure sharing, one individual doesn't have to buy all these receivers. We share the fish data that's pinged at each receiver and gets uploaded into the GLaDOS database. And then every researcher can look for their tagged fish to see where they've been. Yeah.
Why do we have such a density in Green Bay of receivers where other lakes have them a little bit more systematically spread out? Why are ours so dense in northern Lake Michigan?
So part of it, they're spaced actually probably pretty similarly. It's just the sizing of the map. It shows even more just because Green Bay is small compared to all of Lake Ontario or Erie. But they're about 8 kilometers apart in most of the grids throughout the Great Lakes. But and it's nice to have everything kind of spaced evenly, because it's great when it's detected at a receiver. But when it's not, it could be just outside of the detection range, or it could be really outside of the detection range. So by starting to build grids instead of just like, oh, we're gonna put a line of receivers at the mouth of between Green Bay and Sturgeon Bay, then you only know sort of like when they either came in or went out. But then by adding like additional lines, you could start to get that information. So this map is as of like last week with all these. But again, it's been a big evolution in time, and if I rolled it back, even 2024, on the eastern side of Door County earlier in the year, there were no receivers on the eastern side of Door County. They've expanded to answer questions that have been related to lake whitefish. So they keep kind of growing, and again, just we can get into a lot more of the how much time it takes to go and tend each of these receivers, but that's also why you don't see part of Lake Michigan with receivers, or even out in the middle of Lake Superior, because it requires somebody to physically go out to them, pull it up, download the receiver, and go back out, and that requires vessel time and a lot of crew time. Yeah. Yeah, they're all sunk. And the depths that they're at really ranges based on how deep they are in the water. There's been some technology change where now there's acoustic releases, so you can drive the boat over, push a button, and then the buoys, it sends the receiver up. Used to be you'd have to like, use a snag line, catch it, pull up. Some of the receivers are still like that, but once you start getting into really deep water, I mean, that starts to be really challenging, and then trying to have to send divers down is a lot. But this has really evolved seriously from like 2011 to now, a major change. And again, I think this is one of our biggest incredible success stories in the Great Lakes that this region can be so proud of, is the amount of collaboration and what we're learning, unraveling mysteries of all sorts of species. And again, because of the species of, really big concern, the walleye, the lake sturgeon, the lake whitefish, etc., it exists. And people like me who are studying the underdogs, the suckers, can piggyback on all this amazing stuff and start unraveling the mysteries of suckers. So in 2022, I was extremely fortunate to have the financial support of the U.S. Fish and Wildlife Service, Green Bay Fish and Wildlife Office, a couple reps in here, so big shout out to Fish and Wildlife Service. We got 50 transmitters from them. And then in a meeting, somebody from Wisconsin Sea Grant said, hey, I think we can probably buy 10 more acoustic tags. Sorry, I think I said receivers, the tags for the fish. So in 2022, we were able to put 30 in females and 30 in males and start to figure out where they go. And Fish and Wildlife Service folks were there to help me with the tagging. So for the tagging, a little bit more involved, because these tags are larger, a little bit bigger than about the length of a AA battery, not much more wide than that. You only need to make a little incision, just longer than the width of that tag, pop it in the body cavity, do a couple of sutures to close it up. They're absorbable sutures. The fish has a hose of water going into its mouth to keep its gills wet. It's got a TENS pad, which is how many people have used a TENS pad for muscle stuff? We can use it to do an electro mobilization of the fish. It's a quick process. The more fish that you do, and you get faster and faster. But this process is really nice. There's no chemical anesthesia for fish that can allow a surgery to happen where you can release the fish right away. They're working on getting one of the chemicals approved for that, but if you're out there and you catch a fish that's just been tagged and you want to eat it, you don't want to have to worry about making sure that it's cleared that chemical. So these are really great. And what's been very cool too is once we tag the fish with an acoustic transmitter, I also outfit them with one of the inexpensive pit tags, because again, these acoustic tags have a battery life, in this case 3 years. The pit tags don't have a battery, they don't die. When they go over an antenna or buy a handheld reader, there's a little electromagnetic switch that turns them on, and that's how that information is transmitted on the ID. We also put a yellow loop tag behind the dorsal fin of the fish, So if the DNR or if an angler were to catch a fish, it says, it gives an indication that it's tagged, and we try to let people know about it. So also some of the other folks that are collaborating, the study, University of Wisconsin, Stevens Point, Michigan State, Cornell University, of course, I mentioned Wisconsin Sea Grant contributing tags, as well as the support of GLaDOS and the Great Lakes Fishery Commission. And my colleague Chris Vandergoot, who's no longer with us, this picture back in 2022 of us deploying an acoustic receiver that gave me real-time data of when the fish were there. I could look on my phone and see if they were being pinged at that moment. He ingeniously used his croc to slide the receiver under a dock. I love that photo. So some of the results today with this initial 60 fish that we tagged back in 2022 through 2025, we started to see very similar return rates to what the pit tag showed us, very high spawning site fidelity. So in year one, 44 out of the 60 returned to spawn. In 2023, one had died during spawning. It was captured by an avian predator, most likely. How do I know? I found it on shore. The guts were already gone. The only thing that was left was its yellow loop tag, which was still on it, but at least I knew the fate of that individual fish. So then out of the remaining 59 fish, 40 out of 59 returned in 2024. And then last year, before their batteries died, 34 out of 59 in 2025. That real-time receiver was such a treat. Yeah.
So of the 40, is it 40 of the 44, are they the same fish or are they different? You know what I'm saying?
Yeah. So just of the initial, but yes, a lot of that initial these are all, there's only one cohort of fish tagged.
Yeah,
Of
but
that
I'm
60.
thinking like, so the first year 44 came and then
It all,
were they the same 40, was the 40 part of the 44?
a lot of them, yes, but
Yes.
there were some ones that we didn't see one year that we saw another
That's what
year.
I-
Yes.
Okay.
Yeah. But generally speaking, seeing that high return rate, high spawning site fidelity that matched up with what we were seeing with the pit tags. And what was cool, one of the days, we had all 40 of the fish at once being picked up at the real-time receiver that's just outside of the creek, where they were like waiting for that right water temperature to go in. And me working in Chicago, what was really nice too is that real-time receiver gives me the real-time water temperature, so I could start to look at, how quickly was it warming up? That also gave me some indication of how, when do I have to mobilize to start getting up to Door County. This is, a shot year after with one of the white suckers. You can see the yellow loop tag behind its dorsal fin. And what was really cool as well, working in this tributary that in the deepest spot is like a foot, maybe a foot and a half on the deeper water years in some spots, but you could so clearly see what's going on. And we tagged fish one day, took a break of tagging, tagged fish the next day, took a day, and wrapped the tagging up over 3 days. But after the very first day of tagging, we saw fish with the yellow loop tag spawning. So right away, they resume their regular behavior. And that's so important with this kind of tagging because we're making assumptions that the fish that we tag are behaving just like their non-tagged friends. So that's really cool. This is a different way of deploying a listening station in a shallow environment. It's not deep enough to have it sitting upright. So in this case, it's laid on its side. The business end's at the pointy end. So I have it, so it's facing downstream as the fish swim upstream past it, they get picked up. But normally out in all the rest of the stations out in the Great Lakes, they're sitting upright, so they have the biggest 360 radius for being picked up. And in really clear, deep water, a 1 kilometer detection radius, they can pick up, it's a pretty strong signal. But things like aquatic vegetation in shallower waters, if that grows up, or if you put it down and there's like a really rocky area, anything that it's a line of sight signal and transmission. So anything that can get in the way would, you know, slow that or minimize the detection range. And of course, boat noise and other things can temporarily, affect that signal transmission, but those are temporary things. So I'm waiting for this last bit of data. I just got it on Tuesday, because it's a collaborative, process and, you're working with a bunch of people who are out tending all of these receivers and then needing to go through and quality control check and then upload it to the database. I can only see the receivers that I have and pull in until all the information comes. So I don't have the full picture of what happened right up through all of 2025, and even some of the end of 2024. But I can say that all the fish that are in that big noisy graph, what you're seeing is just sort of how long for each individual fish do we have data for. And already can see that for a lot of them, we have quite a bit of data. And so that's really helpful because with more data, it's more of a story. But even for the ones that disappeared out of the grid, who knows where they went? Initially, those receivers, nothing was out on the eastern side of Door County when I started to tag the fish. But what we would see is there would be some fish like this bottom one here. There's a big gap. That was when the fish left Samuelson Creek, went out towards the east, got picked up on the 2 listing stations in the Sturgeon Bay Canal, went out into Lake Michigan, the big waters, and there were no receivers to pick up where it went. Where it went in that time, who knows? Could have gone a long way, could have gone a short way. We had nothing there. But then we also had individual fish that were picked up at a lot of stations within Sturgeon Bay and then out in Green Bay. So already starting to see some repeatable movements within a fish, but fish that are spawning together are not all hanging out together year round. And some are making bigger moves and some are making smaller moves. But we've had fish from Samuelson Creek get picked up up in Fish Creek and out by Chambers Island, as far down south as receivers by Dykesville. And then I just found out too, well, one of the people who is waiting for them to upload their receiver data was texting me at the on the weekend saying, all right, I'm gonna get this in real soon, but here's some previews. I had one of your suckers was picked up on these little mini grids. You see these little groupings of a few receivers south of the Sturgeon Bay Canal. Those went out for like whitefish. Again, the idea is at least, starting to fill in some gaps so that they could start picking up fish if something went south of Sturgeon Bay Canal. With one of the white suckers from 2022 is getting picked up between Algoma and Kewaunee in September 2024, and then back in the winter, and then picked back up in Sturgeon Bay and back in Samuelson Creek to spawn in 2025. I will soon be able to see all of the data. Stay tuned for future fish tales. This is enough of a tease, but that is really cool. But it also shows like how every other receiver that goes out into those areas, it's so helpful to fill in the gaps of where they might be. Great, then fast forward another 2 years, we added another cohort of white suckers being tagged, this time in Fish Creek, a tributary directly into Green Bay to see. Now we've got this whole grid of receivers out there versus in Sturgeon Bay, we've got a number of receivers through Sturgeon Bay and in the canal, but it's not like a grid. So what are we gonna find? So we were able to tag 29 suckers, 15 male and 14 female. What was really cool here is that in that process of tagging fish, Fish Creek, right by the highway, lots of visibility to the public. Peninsula State Park is right there. Lots of people from the public coming and asking questions. Oh my gosh, I'm talking forever. Sorry. So there was, what was really cool too, is that we had a lot of communication. Again, US Fish and Wildlife Service Green Bay office helping me tag. Josh, who's in the picture, is in the audience. Wave to everyone. So what was really cool, a moment from this year that I won't forget, we had tagged the fish. I was like being protective mama coming out day after day, like hoping people weren't going too much into the creek and handling the fish, because a lot of people do like to catch them, but I would just want to let them know if they saw any with yellow tags in their back to handle them with care. They had just had surgery. So I come across like a dad who was on the bridge, and I was like, "Hey, how's it going?" And he's like, "Good." Stranger, why you're talking to me? Like, "Oh, I just wanted to let you know that we put some acoustic tags on some of the suckers in Fish Creek, and they have yellow tags in their back." He's like, "You're putting acoustic tags and suckers?" He's like, "I know about all the really cool tagging and tracking studies that's happening with smallmouth bass and the lake whitefish and that." He's like, you're spending that money to do that on suckers? That's cool. Kids, get out of the water, leave the suckers alone." And so for me, that was like a really cool moment, that investment in that technology to tag the fish has suckers swimming in the big leagues with like these other recreationally important species as well. And that's been really cool. And so some of the information today, again, don't have tons aside from the return rates. Out of that initial 29, I know for sure 12 returned back. I had trouble getting the receiver close to the mouth in before some of the fish started to return. And I had one of the receivers way up in Fish Creek above the culvert on the other side of the highway. Not all the suckers were necessarily gonna go that far upstream, but it's a tricky spot to put these in. So that was a good spot for me to put it in one of the receivers upstream of the culvert, but I would have probably missed some fish that year. But this just shows, these blue lines are periods of time when a number of the suckers were being picked up. Well, one thing that happened not long after one of the fish, like once we had done the tagging, one of the fish went from Fish Creek, was getting pinged on a number of receivers, heading down towards Sturgeon Bay, getting picked up on receivers in Sturgeon Bay, and moved all the way down by Samuelson Creek. So just like we were seeing fish from Samuelson Creek going up by Chambers Island and Fish Creek, that movement of some suckers down that, from Fish Creek this way too, that's pretty neat. So still more to come with that. And then last year, we were able to expand the acoustic telemetry study into another species, long-nosed suckers. So I mentioned it wasn't until 2024 when the acoustic receivers went on the eastern side of the Door County Peninsula. Most of the creeks that have long-nosed suckers are on the eastern side of the Door Peninsula. They're not on Green Bay side tributaries. So that meant that we could tag long-nosed suckers in Hines Creek, which has been a part of my volunteer monitoring program since 2017. So this time with funding from Door County Land Trust to buy the transmitters, field support again from the amazing Fish and Wildlife Service Office of Green Bay, we were able to outfit 15 males, 15 female long-nosed suckers with tags. And similarly put the yellow loop tags in, similar to the white suckers right away. You release them, they started to get back to business spawning. One of the days I was out with an author who's writing a book about fish migrations. Like, we're talking the Pacific salmon grouper, all these like well-known, well-celebrated migratory fishes. He's gonna have a whole chapter on suckers in there as well. I got him in the water taking video. And I'm taking video and the suckers are coming between my legs and they're spawning and hitting my boots and I've got video and I'm squealing. It was just really great to see. Maybe you can see in that picture, that one male long-nosed sucker. All the males for long-nosed suckers get that red bar, by the way. Can you see the one near the top with the yellow loop tag? Again, even just with a GoPro and being able to see them like that, you know, fish wasn't afraid of me after he was abducted and tagged and let go. So stay tuned. This year, we will be able to see how many of those fish come back. And we'll find out. I started to get some little snippets of where some of these long-nosed suckers had been venturing off to by getting, the DNR office let me know that on November 19th, they encountered one of the tag long-nosed suckers in a netting survey they were doing for whitefish. They saw that it was one of the tag fish. They released it, but it was caught just off of Jacksonport, so not super far from where Hines Creek is by Bailey's Harbor. But also the Stevens Point person let me know that in the mini grids, that go all the way down by Port Washington, we had one of the fish being picked up down by Port Washington, as well as one being picked up Manitowoc and Sheboygan area. So having those receivers in that area, so key. So I can't wait to see who comes back and what other adventures they're having along the way. And with additional support again this year from Door County Land Trust, we have an additional cohort of suckers to tag another 30 fish that will do an even split between males and females with the amazing Fish and Wildlife Service helping me. So lots more to reveal, but the big takeaways, as I've kept you captive for over an hour, people really connect with stories of movement. So the tagging and tracking, whether it's been the pit telemetry or the acoustic tagging, is really helping people to connect to these native species that just aren't game species, but they're just as cool. So being able to tell the stories about the males coming into the creek at night and doing these reconnaissance trips to see if the ladies are there. That's cool. Being able to talk about the adventurous fish that went from Samuelson Creek and Sturgeon Bay to be hanging out and spawning and living it up in the Peshtigo River. Cool. These movements of fish from Fish Creek into Sturgeon Bay, these adventurous fish that are going way down to Port Washington and then coming back up, like, that's awesome. I've done quite a bit of outreach. Obviously, these kinds of talks are awesome. But also being out by Peninsula State Park, interacting with guests during spawning season has been really cool as well. I already mentioned Ben Goldfarb, who's a really great environmental writer. He wrote a cool book about beavers called Eager, one about road crossings, and now this one that he's working on for migration, that he'll put suckers in the same pages of other celebrated migratory fishes. So everything we're learning, just stay tuned, more stories to tell your friends, get involved, and I think that's it. Oops. Yeah. So, email, if you wanna contact me, I'll leave that up for a minute. Hopefully I was a good person and also brought some business cards if somebody wants to reach out, but I think it's question time.
Questions?
Yeah, these receivers that you're make sure everybody hears, including the people. Okay, these receivers that you're putting out there in the bay and the lake, do they have to be serviced at all?
Yeah, every year they get out, their service, they're pulled up, have the information downloaded and their battery changed to make sure that all year round they're able
Markers
to.
for them, the buoys out there on them, or how do you know where they're at?
The people that set them out have the GPS locations, but the whole point is that they're not something that's gonna disrupt boaters or netting activity, etc.
So like something that's 50, 60 feet deep, they have to somehow lasso that thing and.
Now, I mentioned that for the deeper ones, they have an acoustic release. So this orange box that you can see in that second picture there, that works to talk to the receivers. So you can send a signal from that to the receiver, and it sends up a buoy with the receiver on it. They can change hardware and send it back down. But for some of them, and certainly before that technology became, prevalent, the whole thing was using, an anchor with a snag line and being able to pull it up that way. It's a lot of work, even with the acoustic release, a lot of work. So it's a very serious time commitment. And then, of course, having vessels to get out, to these more offshore areas.
That's why you saw fewer dots in the deepest water.
Yeah. But even what's happened, dotting the shoreline even too around Lake Superior, even to be able to pick up those kinds of movements, I think what we're gonna see for Lake Michigan too, there's a lot of people, again, just collectively, all the states and the agencies talking about how, at least if along the shoreline, we could have basically that whole perimeter with some receivers. It's not as far to go out, but for all the fish that might swim along the shore, that would be really beneficial. But yeah, it's a lot of work. So having all of Lake Erie, Lake Ontario, Lake Huron gridded out fully, that's major cooperation between so many agencies, to get that.
More questions?
Oh, so many.
I'll go over you this time.
And also, if you have to leave, don't feel bad to get up. I talked a lot, so.
My question is, because the water levels are down, is this gonna cause a problem for tagging?
I think, so the bigger thing for the suckers is just you never know exactly what date they're gonna come in. Things can change, every year is different, right? So they might have the right water temperature cue to say, yep, it's time to move in. But then you could have a big cold front come through and the temperature drops. Depending on the tributaries, that can either mean some of the fish just move back out and don't come back in the spawn, or that some are fine. It's been really interesting to see this over the years. Water level itself, yeah, if you don't get the good snow and that runoff and the rain and the water level is low, what we were seeing with all that 7 years of data of matching up, what the water temperature and the flow cues were on years when the water levels were lower and water levels dropping faster, they tended to move in at a slightly lower temperature. So that can have more of an impact of just when they show up, but as long as there's sufficient volume of water for the fish to come in, we're pretty good at catching them and getting them tagged, but.
So have you tried to calculate whether the population is growing or is it, I mean, where are you statistically on this now?
So this doesn't really get at a population estimate. It would require a lot more effort, but to really catch that, but I can say anecdotally, there's a lot of information both from I've heard both from folks about the Great Lakes Indian Fish and Wildlife Commission near Odena, Wisconsin, as well as some folks in Ontario, that particularly during the spawning runs, that when you see them move into the tributaries, a lot of the things that I'm hearing is, oh, the suckers used to be so thick in this river, you could almost walk across their backs. We don't see that anymore. So these are the things that are concerning. Like, we don't really have like a good population estimate. We could be losing some of these species, the numbers, and it's happening under the radar because we're not paying attention. So I think.
But at the same time, because you have a high return rate, it does seem to indicate that the mortality is not real high.
The mortality, yeah, the mortality doesn't seem to be high, but again, this is still some snapshots of. But it's not really a population estimate for sure.
Any question on this side?
I think you have to touch this on the, as well.
Oh, okay.
I was just curious if you felt those antennas were strong enough to pick up a tag if it ended up in an eagle's nest.
Oh, no, so the acoustic telemetry really only works well underwater. Radio tag though is a different story, and that's another way you can tag, and you could use an antenna, but they're a totally different setup with an antenna that has to be typically outside of the body of the fish, and they don't work as well in deeper waters to use like an antenna to listen for those fish. That's why we've gone acoustic. Um, if you had a fish that was preyed upon and taken to a nest, no, you wouldn't know unless you found the tag. But that is, again, like I did see that one fish that I'm sure was an avian predator, and just happened that I was there and saw the carcass. Some really cool, interesting things, and as we were catching some fish in Fish Creek, we did see some fish with talon marks on their backs. And one of my volunteers got really cool footage of an eagle landing on a sucker and riding it in the creek for a minute, and they didn't quite get it. And then it takes off, but then later, sure enough, there's a carcass. So that was also part of protected mummy, like after tagging, I'm like, oh, please don't let it be like an eagle or an osprey that's got one of my fish on the shore.
More questions?
So in the non-breeding season or spawning season, are the fish deep swimmers, the suckers, or are they generally shallow?
So they've been caught in all depths too. So again, getting some survey data from Fish and Wildlife Service, and then tomorrow meeting with DNR to find out where they've been capturing the fish. They are finding them in both shallow and deeper waters as well. But how much is an individual moving at a time? This is where we'll be connecting the dots. So what's been interesting since the zebra and quagga mussels really being established throughout the lakes, that's moved a lot of nutrients from offshore areas to inshore. So one thing with the suckers, if they're making movements from offshore to nearshore areas, that would be having them acting as energy conduits, feeding in one area, pooping in another, moving nutrients around that way. So that could be another ecosystem service that they're providing. So,
You got another one?
Yeah, my last question.
yeah,
So in the male, female tagging.
yeah.
The return rate, have you noticed that it's the males who are always coming back and the females missing, or the other way around?
It's been both where we've missed seeing some of them come back. And we've had then both, one, the Peshtigo River, and then the Big Creek one was a female, one was a male. So it seems to be that straying and adventuring different places is a mix. But when it comes down to potentially taking a year off from spawning, it would make a lot of sense if a female does, because for the amount of energy that they have to put to make eggs is significant, because they put a lot of fat, the lipids, into their eggs so that the little baby fish, as it hatches before it's ready to feed on things, it's got a yolk sac to feed off of. So it takes a lot more energy for the female to make her eggs than the male for the sperm. So
I'll come back to this side. Any questions?
Yeah, I have a question as a follow-up to that. Because of that energy level, do the females tend to stick closer to the creeks where you tag them?
have not dug that far into the data. It's right now, it's just been more of just seeing a lot more individual variability. I'm hoping just as a whole with both combining the Samuelson Creek and the Fish Creek white suckers, pooling those numbers, because again, it's still, yeah, like 89 fish. That's still a decent sample size. But the longer time you have tracking data for one individual, it puts more of a story together. So I still have a lot of digging in the data to do for future fish tails. It's just a good way to make sure that Mark has me back. But yeah, I think right now it just seems more of a mix of both. You know, there's individual variability that we see for both the males and females, but I think that, that could be something that does end up shaking out. I don't know, we'll see. A good thought.
We regularly take water temperatures in the NP. I was wondering if you remembered.
No, give me,
I checked AI and it said 43 to 50 degrees.
yeah, I think it was 43.4. So what was interesting, yeah, for Fahrenheit,
Not the state.
somebody can fact check me in a second from my own site. It is 43.4 Fahrenheit. And what was interesting, because in the little bit of literature that was out there, we were seeing that it's actually a lower water temperature than what the literature is saying. But the benefit of having the citizen science aspect is there's only one me and lots of not me. So all the volunteers that are, the eyes and ears on the ground at all of these tributaries across the latitudinal gradient, peeking in the creek. Are the suckers here yet? No. The suckers here yet? No. Oh, here they're here. We can mark that in, match it to that water temperature cue. Seeing it lower could be a couple things. It could be just that having that higher resolution of data, finding that it actually was a lower water temperature that they were migrating into, or we're already starting to see some of the impacts of climate change and they're moving in earlier and at a lower water temperature.
So cold.
Yeah, it is. It is still chilly.
Are you worried about data centers taking away water from sucker fish for all the AI users?
Yeah, I mean, that's a fair question overall. I mean, I think that the thing with suckers is their ability to have the free will to move around, like any of the fish can find the habitats, but I think I'm not worried about it from that aspect. Maybe other things, but less so for suckers having places to go.
With all these different agencies from two countries, how good is the exchange of information? And I'm just amazed you've got such a big program together.
Yeah, it's been a lot of effort, but every year there's a meeting that folks from both sides of the border come to get together and discuss, like, yeah, how do we keep working together to fill in gaps? Of course, Lake Michigan's just, within the United States for figuring out some of those questions. But it's an incredible, it's so collaborative, sharing ideas on best deployments and retrieval of the receiver. I've been involved in a lot of the best tagging practices over time, whether you're tagging fish from a boat versus shoreline. What's the best suture material? Like, what's the, switching to using electroimmobilization, because there's no chemical anesthesia for the fish to have to recover from, and all these best practices. That information's really well shared. And again, it's just such a success story. Fish nerds unite and work really well together.
And actually, we need to say Great Lakes Fishery Commission. So the agency that is in charge of controlling sea lampreys and helping coordinate things, they're the ones that are the basis of the whole GLAVIS. So the whole structure that's been set up has been through the Great Lakes Fishery Commission. And the fact that addressing any fishery issue in the Great Lakes is a multi-agency approach.
Yeah.
And so the whole atmosphere or whatever of the agencies is a cooperative thing to begin with. And that's the coolest thing about the Great Lakes.
Yeah.
Yeah. Yeah. Yeah, you have to leave, go ahead. These questions are great.
And then I think we might have to check online to see if there's any questions.
Yes.
I just kind of wanted to double back to, how did you get going on the sucker initiative? Because I think we as a society put a lot of emphasis on commercial fishing.
Yep.
Sport fishing, you say fishing nerds, but we're not all fishing nerds.
Totally.
There's groups that look at suckers, carp, sheephead, white perch, all crash fish, and are competing against the fish that, the fishermen want to catch, whether it's walleyes or perch or northerns, we kind of rankle and stuff. We don't put a lot of emphasis on our native species. Is that how this program got going, that you wanted to start looking at protecting native species, non-sport commercial fishing? How did your program get started? Because it's not really that old.
Yeah, so I started at the aquarium in 2016 and I came from working with an amazing saltwater species called bonefish that anglers go nuts for, and I never had trouble getting an audience to hear me talk about bonefish. Not that I've had too much trouble getting people to listen to me talk about suckers, but yeah, that clout between like the sport fish versus the non-game fish. Coming to work at the aquarium, the whole point, I'm a team of one for the migratory fishes program. It's me. And then I collaborate widely, but the opportunity to fill in a gap that wasn't being investigated by other researchers is really where, that opportunity came in to work with suckers. And Pete McIntyre, who is faculty at University of Wisconsin-Madison, had already done a bunch of sucker research, and it worked to be a good species to start with for that reason. I wasn't going to be duplicating any work. I was going to be filling a void for a need for data, and I love them. They're really cool.
So there is one question
Yeah.
in the chat box. Do you find any relationship between high number of sucker spawn with macroinvertebrate volume going up and high numbers of spawn equal more macroinvertebrates?
I don't do surveys of the macroinvertebrates, so their overall success rate, I imagine that it's more to do with the growth of those individuals that are there. I don't know of the numbers.
But the energy from the eggs do support the macroinvertebrate.
Oh, absolutely. But whether it's the numbers change because it's a snapshot, it's a period of time that they're in there. And those macroinvertebrates are there. Of course, it could benefit them just from the fact that they can grow bigger. They could end up being more successful.
Okay,
But
so
yeah,
if there's any other question from the people online, if you unmute yourself and talk, I think we'll be able to hear you.
and also thanks for hanging out online if you've been here the whole time.
But hearing none, there's not many advocates for a single species like Karen.
But it's not just a single species, it's white suckers.
Group of fish. And I think if we actually had a congeniality award for all our fishtail speakers, it would be hands down Karen Merchant. So, thank you very much, Karen, and let's give her a big hand. February 12th, it's walleye. See you then.
These are recorded,
Yeah.
so you could go online and at a later date.
We take you out of school on Friday.